Gaurav, are you there? Gaurav, I'm audible to you?
Yes, Kumar.
Gaurav, now we made this session live on YouTube also now. Okay, so we'll start the session by sharp after 2-3 minutes. Okay, let's wait for another 2 minutes more.
Definitely, no problem.
Yes. Gaurav,
Yeah, Kumar.
I think we should start now. So we can just begin the presentation by 3 o'clock. We have enough attendees to proceed.
Yeah, so you will start the introduction?
Yeah, we need to start right away only because we have enough attendees to proceed, and we can begin on the PPT by sharp 5 o'clock. So sharp 4 o'clock. Sorry.
Okay, so you can start.
Yeah. So welcome everyone. Thank you for joining us for today's webinar. My name is Dr. Kumar Jitendra, and I'm the Chief Editor for Microbiology India, the leading publication for pharma and analytical magazine. So today we are presenting very interesting session on microbiology quality control techniques in pharmaceuticals and biopharmaceutical industry with association with Abdos Life Sciences. Is a leading manufacturer of centrifugeware, laboratory equipment, battery dishes, pipette tips, beakers, and measuring cylinders manufacturer. So just a little housekeeping before we get started. If you have any questions during the presentation, please type them up into the question box showing in the left side of your screen in the control panel, and I will bring them up just after the presentation during Q&A session. So now without further ado, we will turn the time over to our session. Our presenter today is Nimesh Desai. Is a quality control leader, deputy manager in quality control in Alembic Pharmaceuticals, and other speakers is Mr. Gaurav Sarda, is a product and marketing manager of international business in Abdos Life Sciences. So, dear all attendees, please make your audio muted. Dear all co-organizers, please also make your audio muted to make this session more and more successful. So now I'm over mic to Gaurav. Gaurav, kindly proceed with the session.
Yes, thank you, Kumar. Good afternoon, good evening, everyone. It's a pleasure to welcome all of you here. Myself, Gaurav Sharda, Product Manager, International Business from Abdos Life Sciences, and I'll be talking about today's program. And I would like to talk first about Abdos and what it's all about. So coming to this, Abdos has been divided into 5 groups, major groups: distributor, FMCG, contract manufacturing, multi-layered laminated tubes, and life sciences. It was established in 1967. It's a 54-year-old company, and Abdos Life Science was founded in 2009. One of the landmark achievements which we achieved was a production capacity of 400 plus million filter tips annually, and we have a team of more than 1220 dedicated persons. We believe in consistent and high quality product which comes naturally from our company. Abdos was established in 2009 with a state-of-art facility at Roorkee, Uttarakhand, and we have our offices in Delhi, Kolkata, and European hub, which is the warehouse facility in Netherlands, Venlo. We have a dedicated team of over 400 people in the life science division itself. Abdos division vision is to be the first choice of customers by building long-term relationship with them through our product range, service levels, innovations, and sustainability endeavors. And the missions which we have mentioned here aligns with our vision. In 2022, Abdos wants to touch the lives of people by ensuring, empowering health and hygiene across all the world. So our long-term vision is to grow aggressively, consistently, and maintain integrity with building long-term relationships. So these are certain pillars of Abdos. We are one of the largest premium manufacturer of plastic labware in Asia. We have over 400 products in our catalog, and we cater around 65 countries globally. We have been certified with 21 quality certificates, and all our products are cited in leading research journals. We have a state-of-art manufacturing which is made by in India itself, and it's spread across 40,000 square meter campus, and it's been validated with ISO Class 8 clean room. These are certain pictures of our plant and clean room facility, which has been validated with ISO 8. You can see all the pictorial representation. Automation, robotic platforms of Abdos ensure there is no human interference, and we can ensure this consistency by providing an endotoxin level of less than or below 0.005 EU per ml. So we ensure there is no human interference, and thereby reducing the carbon footprint itself. We have our in-house R&D team, which provides custom solutions and for specific requirements for our customers or end users. Abdos has recently launched its Generation 2 pipette tick box, and we have successfully filed this patent, which is being powered by its anti-warpage and anti-shrinkage technology. And Abdos is going bigger. Our campus, we are adding around 1.5 lakh of square feet area, which will threefold our production capacity, and we'll get into the production line expansion of certain products. So the Abdos goal is to double their revenue by 3 years. We have our warehousing capacity in other continents like Netherlands. We have manufacturing range of PCR products, biobanking, and biocoded products. We are the largest manufacturer of centrifuge tubes in India and consolidate as the highest manufacturer of petri dish. We are going for the green manufacturing and biodegradable product lines, and we are setting up manufacturing outside India and also expansion of our existing plant. We are also the largest manufacturer of filter tips in India. These are certain quality certificates which we have got, like heavy metal test report, endotoxin and ATP free test certificate, migration test certificate, certificate of analysis, and technical data sheet, which is already available in our websites. These are certain more certificates at a glance. These are the quality management system certificates, certificate of compliance, manufacturing of plastic labware for clinical and medical use. We have been certified with in vitro products, and also we are the European Union representative. These are certain new certificates which got recently: environment management system, forensic DNA grade, and we have been certified with in vitro products for most of our product range. Our international presence is over 65 countries across the globe, and we have a warehousing facility in Europe, Netherlands. So thank you for this presentation. I would like Mr. Nimesh Desai, our valuable speaker for today, to proceed. Thank you.
Yes, sir. Your screen is visible. You can proceed it on. Sir, unmute your audio. Nimesh, sir, unmute your audio first. Nimesh, sir, if I'm audible, please unmute your audio. Yeah, now it's fine. Please proceed, sir.
So, today I will going to explain the importance of microbiological quality control in the pharmaceutical industry. So first of all, your brand is the greatest asset and your greatest risk. So what the quality control is an essential function of the pharmaceutical industry. Drug manufacturer must thoroughly test materials, process, equipments, techniques, environments, and personals in order to ensure the final products are consistent, safe, effective, and predictable. Undoubtedly, a company suffers enormous damages when a drug product is recalled. In universal, in the whole world, there was whatever pharmaceutical industries are following some rules and regulations, and they are following some standards which is applicable to whole world. So here there are three major pharmacopoeia throughout the world, including the United States pharmacopoeia, the European pharmacopoeia, and the Japanese pharmacopoeia are followed most of the industries. Moreover, in Indian Pharmacopoeia is the official book of standards for drug, included therein the term of the second schedule to the Drug and Cosmetic Act 1940. So as to specify the standards of identifying purity and strength of the drug imported, manufactured for sale, stocked, and exhibited for sale or distributed in India. International also there was one international conference on harmonization of technical requirements for registration of pharmacopoeia for human use. That is the ICH is working to facilitate international harmonization in the requirements for the pharmaceutical industries. So here I am going to explain about the pharmaceutical microbiology laboratory techniques. So there are a few techniques, and followed by these few techniques, we are going to perform few tests in the microbiology laboratory. So one by one, we can go through in the depth. So regarding the techniques in microbiology, first of all, it's a spread plate techniques. What is the spread plate techniques? The spread plate techniques involved using a sterilized spreader with a smooth surface made up of metal or glass to apply a small amount of bacterial suspended in a solution over a plate. The plate need to be dry and at room temperature so that the agar can absorb the bacteria more readily. A successful spreaded plates will have a countable numbers of isolated bacterial colonies eventually distributed on the plates. So in this technique, how can we follow it? So to viewing this diagram, what we can do in the technique, we are taking the agar media in the 90 Petri plates. After that, we inoculate the cultures by using the L spreader or triangle spreaders. We evenly spread the suspension on the agar media. We just give the contact time. After that, we incubate it, and after certain time of incubation, we observe the growth of colonies. Uh, what in the past? What is the difference between past and in future microbiology laboratories? So in past, what there was a lack of inventions. We are using glass spreader, glass Petri plate. We need to autoclave it. We need to prepare the media in house. Everything. So it will take a longer and longer period of time. So in current scenario, to achieve the to reduce the time and achieve the highest productivity in market, there was a many scope is open. So right now we instead of glass Petri plate, plastic Petri plates are available in the markets. Also instead of glass spreader, we can utilize that as spreader. It's a plastic made which is pre-sterilized gamma radiated ones. Technique is the pour plate techniques. What is the pour plate technique? Pour plate method is usually the method of choice for counting the number of colony-forming bacteria present in a liquid specimen. In this method, a fixed amount of inoculum from a broth or sample is placed in the center of a sterile petri dish using a sterile pipette. Molten cooled agar is then poured in the petri dish containing the inoculum and mixed well. After the solidification of the agar, the plate is inverted and incubated at 37 degrees centigrade for 24 to 48 hours, or whatever the temperature and time required depends on the pharmacopoeial requirements. So it is a very simple technique. What we can do in that, we take an empty petri plate, we inoculate the samples. After that, we cool down, we taking the malted and cool agar media. So the pouring temperature of the agar media should not be more than 45 degrees centigrade. So by using IR gun and everything, we can measure the temperatures of the agar. We pour it once. After pouring, we solidify it and incubate at desired temperature for desired time duration for the growth of microorganisms. Another technique, that was the membrane filtration technique. Membrane filtration is a technique for testing water samples or sample which is easily filtered out through the 0.45 microfilters. In this procedure, water is drawn through a special porous membrane designed to trap microorganisms larger than 0.45 microns. Afterwards, the filter is applied to the surface of endo-agar plate or any other plate like R2A, SCDA, whichever is the required, depends on the product requirements. And after that, we incubate the plate at in desired temperature for desired time durations. In these techniques, what we can do, we are using that membrane filter, and by using membrane filtration assembly, we keep that membrane into the assembly. First of all, we pre-weight the membrane by using the solutions like 0.1% peptone, and after that we are filtering the product which is easily filterable. So after filtration, we give some desired washing and we take that membrane and transfer onto the agar media. So after transferring, we incubate the plate at desired temperature for growth of the microorganisms. That there was another technique, that was the most probable number. The most probable number is a statistical method used to estimate the viable number of bacteria in a sample by inoculating broth in tenfold dilution, and is based on the principle of extension dilution. It is often used in estimating bacterial cell in a water. But what the pharmacopoeia suggests, that most probable number, its MPN method, is not advisable as compared to membrane filtration or pour plate techniques, because the consistency of the result is not good to compare with that spirit plate or membrane filtration or by pour plate techniques. So it is not advisable method to utilize for the routine pharmaceutical testings. After that, serial dilution. Why it is important in pharmaceutical microbiology? It's a serial dilution is a series of sequential dilution used to reduce a dense culture of cell to a more useful concentration. Each dilution will reduce the concentration of bacteria by specific amount. So by calculating the total dilution over the entire series, it is possible to know how many bacteria you started with. So in serial dilution, what we can do, we are taking that 99 ml of the saline solutions or buffer sodium chloride peptone solution. By use from dense solution, we are transferring 1 ml from first dilution, followed by serially diluted up to 10 to the power 6 or 7, when up to when our desired count was achieved. Why the serial dilution technique is most important in pharmaceutical? Because whatever the testing we are following for a pharmaceutical purpose, there was some specific criteria to challenge the microorganism. In any most of the tests, we need to challenge not more than 100 CFU microorganisms. So if the culture is dense, so there was many microorganisms are present. So from that, we need to dilute that, and we need to achieve the solution which having the number of CFU from 10 to 100 CFU only. So that's why the serial dilution technique is most important in the pharmaceutical industries. So these are the techniques we are following. But what kind of test we need to follow in the pharmaceutical industries? So right now we go one by one. What are the tests utilized in the pharmaceutical microbiology? First of all, the growth promotion test. What is the growth promotion test? The growth promotion test is very important in quality control function in the pharmaceutical industry. It is imperative for establishing the nutritive property of the microbiological culture medium that will be used in a pharmacopoeia's procedure, such as test for specified microorganism. Culture media or media refers to a liquid or gel substance that is used to support the growth of the microorganism. So it is the confirmatory test of the media, whatever the media we are utilizing for analyze the product. So it is the confirmative test whether the media is support the growth of microorganism or not. So by doing the growth promotion test, we can ensure the growth promoting, growth inhibitory, and growth indicative activity of the medias. So the growth promotion test is necessary to ensure that the culture medium is able to support the growth of small number of microorganisms, and that selective medium will grow specified microorganisms. If the new batch of the media is not able to properly support the growth, the pharmacopoeial test for which it is used will fail. So why it is important? For example, the media growth promotion test was failed, and we are utilizing such kind of media for the product analysis. In that conditions, what happens? Whatever the result we are derived by using the false fail media. So the whatever the result of the drug, it may be the questionable, right? So that purpose we need to utilize a media which is passing from the growth promotion test. Why it is mandatory requirement? So to referring the pharmacopoeial reference like European pharmacopoeia, Japanese pharmacopoeia, Indian pharmacopoeia, or whatever, microbial limit test for specified organism, and sterility test. These are the chapter which is harmonized. So harmonization is indicated that considering the all pharmacopoeia, the test procedure was similar. So looking the requirement of these pharmacopoeials, we need to ensure the growth promotion activity of the media. Therefore, growth test must establish the ability of the selective media to meet the three criteria. For selective media, we need to follow the three criteria. First of all, growth promoting property. So growth of the selected microorganism is supported. Second one is the inhibitory property. Organism that should be inhibit on the medium should not grow. And third one is the indicative property, demonstrating that pollen is having a specific morphological characteristic. Why it is required? Because in considering the pharmacopoeial reference, there was a chapter test for specified organism. In that, we are challenging the different microorganisms like Escherichia coli, Pseudomonas, Salmonella, etc. So considering the specific microorganism, there was a specific media are available. So by utilizing that specific media and doing these different activity like inhibitory, growth promoting, and indicative, based on that we conclude the results on the drug product, whether the pathogenic organism is present or not. So here is the just flow was given how to perform the growth promotion test. What we can do in the growth promotion test, whatever the media we received or in-house prepared media, we are taking the plates, we are spreading the culture of different microorganisms which is recommended in the pharmacopoeia. We give the desired incubation condition. After completion of incubation, we are observe the growth of microorganism. For selective media, we are just qualitative test. and for whatever the media, general purpose media like SCDA, R2A, we need to quantify it. So in that condition, we are quantify the CFUs. So there was some specific criteria for quantification. The result should be within a factor 2. That means it indicates that, for example, you are challenging 20 CFU. So the whatever the result we are getting, it should be within 10 and 30 only. So in that way, we are performing the growth promotion test and we approve the media for further product analysis purpose. Here microbial limit test. Microbial limit test designed to perform the qualitative and quantitative estimation of specific viable microorganism present in samples. It includes tests for total viable count and pathogens like Escherichia coli, Salmonella, Pseudomonas, and Staphylococcus aureus. So microbial limit test can perform by different method like spread plate method, pore plate method, and by using the membrane filtration method. So here is the few diagrams are present which indicate the diagram number A is indicates a technique which is a pore plate technique they are utilizing. In diagram number B indicates a spread plate technique, and diagram number D is a membrane filtration techniques. In that way, different techniques are utilized for performing that different tests. Factor for consideration when performing a risk-based assessment into objectionable organism are summarized below. For example, in microbial limit test, what product testing are mainly, why we are doing the product testing in the pharmaceutical industries, because whatever the product we are manufacturing a drug and it is utilized to cure the patient, right? Suppose if there was some objectionable microorganism are there, so it may adverse impact to the patient who is consuming the medicines. So in that condition, suppose instead of these 4 pathogens, we got some objectionable microorganism, then also we need to follow some risk-based approaches. So what we can do, we can follow that the characteristic of that microorganism. We need to see what is the characteristic of the microorganism, the number of microorganism present, the nature of the product as in whether or not the product support the growth, whether the product has adequate antimicrobial preservatives, the use of the product due to the fact that hazards varies according to the route of administrated via the eye, nose, or respiratory tract, the method of applications, the presence of disease, wounds, or organ damages, the intended recipient as the risk may differ from nanoids, infants, and the elderly, and the use of immunosuppressive agents and corticosteroids. So here we explain that microbial limit test. Now the another test was there. It's a bioburden test. When the bioburden test is utilizing in the pharmaceutical industry, the bioburden test in general is often seen as the test which is performed on the bulk solution used for the parenteral products, either manufactured aseptically or terminal sterilized. So it is the just pre-ensuring of the test. Whatever the product we are manufacturing and we claim as a sterile. So prior to sterilization, we ensure the bioburden level of that bulk solution. For that, we are performing the bioburden test. In bioburden test, what we can do, we are taking the solutions by using same membrane filtration techniques. We pre-wet the membrane, followed by we filter out the products. After that, we rinsing the membrane using the 0.1% Peptone solution or desired diluents. After that, we take the membranes, transfer on the desired media and incubate it. We can give the 2 incubation here. First of all, 20-25 followed by 30-35. It is support to both kind of microorganism like fungi as well as bacteria. And in that, after completion of the incubation, again we count the number of CFUs, and we can see that how the bioburden is present in the bulk solution. So it is the convention method, and it is generally utilized in the all microbiology laboratory. But in current scenario, there was some advanced technology in the market. So there was automated detection of the growth also available. Normally, growth is detected by conventional method. Normally, the growth is detected by incubating the agar plate for 3 to 7 days and then assigning microbial growth by the human eye. Thus, using an approach by which the microbial growth can be detected earlier is a good alternative, and validation work is less complicated. So, there was some fluorescence technique was available. So, by using that technique, uh, we we can get the result within a shorter period of time by using some dyes and everything. So for after that, there was another test. It is the most critical test in the pharmaceutical industry. That is the sterility test. Sterility test is designed to demonstrate the presence or absence of extraneous viable contamination microorganism in parenterals designing for human use. Whatever the injections we are manufacturing, we need to perform the sterility test. Why it is mandatory test, and why it should be no there should be a no growth of microorganism, because parenterals, everyone should know, the drug was directly penetrated through intravenous or intravenous. In that way, it's directly contact to the human blood. And suppose there was some contamination in the injections, then it directly adverse impact on the patients. So that's why the sterility test is mandatory in the parenteral preparations. So how can we do the sterility test in the mammalian filtration? First of all, there was a two technique for sterility test. One was the mammalian filtration technique, and another one was the direct inoculation technique. When we are utilizing the mammalian filtration technique, when the product is water soluble and it is easily transferred to the 0.45 micron filter, then we can utilize the mammalian filtration technique. But some of the product which is not able to filter out through using the 0.45 micron filter, in that condition we need to go by the direct inoculation techniques. So in sterility test, how can we perform the test, what kind of media we are utilizing it, and what is the incubation condition and duration of the incubations, right? So in the sterility test, first of all, there was a two type of media utilized. One was the SCDM, soybean resin digestive medium, and another one was the fluid thioglycolate medium. SCDM is to support the growth of the aerobic microorganism as well as the fungi and yeast, while the fluid thioglycolate medium is to support the growth of anaerobes, that is anaerobic as well as aerobic microorganisms. So that's why we are utilizing these two kind of media. So how we perform the sterility test? In sterility, same technique, we are using that membrane filtration technique. In sterility, there was a two type of method is available. One was the conventional method. What in conventional method? It's a one kind of open method. So in that, what we can do, we taking that filtration assembly, we just keep the membrane filter, prevent, transfer the products, filter out the products, wash it, and after completion of that cycle, we pull out that membrane, and we cut the membrane in two equal portion. One portion was inoculated in the FTM, and another one was in SCDM. So it's a conventional method. But in current scenario, the technology was advanced, and there was a closed system was available in the market. So closed system is nothing; it's a canister-based system. So there was a such kind of designing of the canister. So directly the product was filtered out without touching, without human interventions. We can filter out the product. We fill the canister using SCDM and FTM, and in that way we perform the sterility. So once we fill the canister with FTM or SCDM or transferring the membrane from half of membrane in SCDM or FTM. So after completion of this activity, we incubate the SCDM tube at 20-25 for 14 days and FTM for 30-35 for 14 days. Why we can give the 14 days their maximum time for the sterility test? Because sterility is a very crucial test for the patients. So there many microorganisms which are a slow grower and it may not able to grow within the shortest period of time like 5 days to 7 days. So we give the prolonged incubation time, it's up to 14 days. So is there any slow grower microorganism is there? So we can easily identify that microorganism. That's why we are incubating the canister or tubes up to 14 days in the case of sterility. After that, another test was there. That was the bacterial endotoxin test. First of all, what is endotoxin? Endotoxin is a pyrogenic substance found in cell wall of gram-negative bacteria. Gram-negative bacterial cell wall have a lipopolysaccharide. This lipopolysaccharide is also called endotoxins, right? And all endotoxins are pyrogens, but all pyrogens are not endotoxins. It's a thumb rule. So, what is the effect of the endotoxin? Why we are performing the endotoxin test in the parenteral preparation? As I said, parenteral drugs are directly given to the patient and it was directly contact to the human blood. So, for example, in the drug product, endotoxin is there. Then there was a possibility to react with the human blood, and in that condition, patient may suffer for fever, headache, vomiting, etc. And in some time, for endotoxin concentration is much more higher, then the blood clot formation was occur, and in that case, death may occur. So that's why the endotoxin test is most critical in the pharmaceutical industries. How to perform that endotoxin test? For endotoxin test, there was a two type of method available. One was the gel clot method, and another one was the kinetic method. So both the techniques are like similar only, but what is the different mainly? Gel clot, there it's a manual one. We just incubate the tubes, invert it, and ensure that the gel formation was occur or not. And by kinetic method, what there was it's a 21 CFR instrument is there, and based on that Beers and Lambert's they by use of some spectrophotometric analysis and quantify the results. So that is the more advantage in that photometric test. So how the endotoxin test was performed in the industry? So there was a horseshoe crab. It's an Atlantic sea animal. So in market, LAL reagent is available. What is the LAL? Limulus amoebocyte lysate. It's a horseshoe crab blood. So what in that condition, what they are doing, they are taking that horseshoe crab blood, centrifuge it, and preparing that LAL solutions. So and there was an endotoxin, standard endotoxin available in the market, control standard endotoxin. By using that control standard endotoxin and lysate, and we are challenging our product, and we give the incubation condition mixture of that solution, and we incubate it 37 degrees centigrade in dry bath for 60 plus or minus 2 minutes. After completion of the incubation, we pull out the tubes and invert it up to 180 degrees centigrade and observe that whether the gel formation was occurred or not. So based on that, suppose the gel formation is there, then it is indicated that the product is contaminated with the endotoxin and the gel formation was not there. Then it indicated that product was not contaminated with the endotoxin. So in that way, we follow the endotoxin test in the industries. After that, one another test we are performed in the pharmaceutical industries like preservative efficacy test. What is preservative efficacy test? Many multi-dose vials. Multi-dose matlab multi-dose in sense, for example, there was one big vial was there like 50 ml and 100 ml, and from single vial we are doing the dosing from multiple times, like within up to month or up to 2 months, so whatever. So in that condition, there was some preservatives utilizing in the manufacturing of the drug product. It's like benzalkonium chloride and extra stuff. So what is the role of the preservative? Preservative role is it's inhibit the growth of microorganism. So while multi-dosing, there was a possibility to contaminate the drug product by using a syringe needle and everything. In that condition, due to the preservatives, product was not contaminated with the external microorganism, and for that purpose we need to perform the preservative efficacy test. So in preservative efficacy test, what we can do, we are challenging the multiple microorganisms in the power of 10 to the power 6, and we incubate that drug product along with the microorganism in 37 degrees centigrade, and followed by, we based on that intervention given in the pharmacopoeia like 7 days, 14 days and 28 days, we are pull out the samples, and we analyze it, and we confirm whether the preservatives are working or not, how we can perform. So whatever the we are challenging the microorganism, just we are analyze it, and we found what the log reduction was differ from initial count to 7 day, 14 day and 28 days. So based on that, we concluded whether the preservative was effective or not. So in that way, we are performing the preservative efficacy test. So after that, in pharmaceutical industry, we are analyzing materials and everything, but there was a two most important things are utilizing. That was the water and EM. So here I just brief about water, how the pharmaceutical industrial water system is working, and how the microbiologists are monitoring that water systems. So in water testing, there was a multiple type of waters are utilized to generate the purified water and source water, purified water and water for injections. So we need to monitor the source water. Source water may be borewell water or river water or pond water. After that, it is passing through the multi-stage system like softener. After that, different size membranes, RO, EDI, and in that way we are producing the purified water. So after generation of that purified water, by using the multi-column distillery system, we are generating the water for injections, and that water for injection we directly utilize for the parenteral preparations, and purified water generally used for the oral the counter drugs. So we need to monitor the water system on the daily basis. So what we can do in that, we are scheduling the water samples. We are collecting that water samples, and we analyze as per that membrane filtration technique. Also, we are performing that specified microorganism test, and in water for injection, we are performing the LAL test, bacterial endotoxin test. Because it is so process water sampling, collect water sample at a crucial point, sample aseptically, collect sample in a manner consistent with the actual use in manufacturing, establish microbial alert and action level based on historical data, use the test method that determines new numbers and type of microorganisms, identifying microorganism if they may be objectionable in a personal care product or any pharmaceutical products. So why it is important to monitoring the water system on routine basis? Because there was a many microorganisms which have the capability to generate the biofilms in the water system. So in that condition, uh, it may in that condition it may the quality of the water was compromised. So that so to consistency of the water quality, we need to monitor the water system on the daily basis. For example, few of the microorganisms like Pseudomonas, Salsonia, Staphylococcus, Methylobacterium, Clostridia, Salmonella, etc. have the capability to generate the biofilms. So from when bacteria are attached to surface in a moist environment, produce a slimy gummy substance, extracellular polymeric substances, resist to chemical sanitizers, release pioneer cells for further colonization, and can proliferate in a very low nutrition content, containing high purity systems. After that, environmental monitoring. Environmental monitoring describes the microbiological testing undertaken in order to detect changing trends of microbial count and microflora growth within clean room or controlled environments. The result obtained provide information about the physical construction of the room, the performance of the heating, ventilation, and air conditioning system, personal cleanliness, gowning practices, the equipment, and cleaning operations. Most commonly occurring microorganisms come from human skin, are gram-positive microorganisms and common environment fungi, which include the following: Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus capitis, etc. Whereas those associated with the eye, ear, and mucus, including gram-negative microorganisms, which can rise on rare occasion directly from the operator and includes the following: Pseudomonas aeruginosa and Escherichia coli. So there was some guidance requirement. What is the limit of that environmental condition based upon their grade size? For example, grade A. Grade A is a critical grade where the injectable drug products are failing. In that condition, uh, first of all, there was a four technique for the environmental monitoring utilized worldwide in the pharmaceutical industry. One was the active air sampling, another one was the settle rate, third one was the contact rate or swab test, and fifth one, another one was the glow print monitoring. Grade A is a critical grade where the limit of the microorganism is less than 1 CFU only. For grade B, grade C, and grade D, here the tabulated form is there. So we need to follow this limit and we need to monitor the environments on specific area where the drug product are manufacturers. So this is about the role and responsibility of the microbiology laboratory in the pharmaceutical industries. So in brief, I say that there was few techniques available, and by using few techniques, we are performing the many tests. Techniques are common for all the tests, so we need to follow that techniques in precautions way. And microbiological testing is a 100% manual analysis, so we need to follow many aseptic practices. We need to take care to avoid the false result while analysis. So what in past? In past, lack of the innovations and everything, we are using then many conventional techniques like glass plate, glass spreader. Even we are using that copper loop. Multiple times we heat it and utilize single copper loop for multiple times. So in that way, but the scenario was changed. In current scenario, everything is available into ready-to-use forms. So by using that ready-to-use forms, we can consuming first of all microbiological times. Second thing, we by using that ready-to-use pre-sterilized materials, so we can avoid the falsification of the result. Also, we are saving our precoces time, and we can give the outputs on minimum time using that ready to use things. So this is the final conclusion. The quality control is an essential part of the laboratory routines. It is not only important for compliance with standard, but also reduce risk to the end users and consequently to the manufacturers. Thank you.
Thank you so much. Thank you so much, Nimesh sir, for this mind-blowing presentation. And really, I'm 101% confident all attendees definitely enjoyed the session. And being a microbiologist, I would first recommend to all of you that in your laboratory workflow, when you start from zero to 100 till your finished product, that microbiology playing an important role for us to make it more and more healthier and to make more and more consumer friendly. And the whole testing, whatever sir explained here, these all testing also depends upon the quality and quality and sterilization of the equipments, machineries, petri dishes, consumables you are using. And today's session is mainly hosted behalf of Abdos Life Sciences, who is a leader in manufacturing of that dishes consumable for biopharmaceutical consumables, petri dishes, pipettes, measuring cylinders, and all. So without any further ado, just shift this session to another step, that is like Q&A section part. Nimesh sir, we have some sort of question here. The first question is, what if there is a viral contamination? Like you have explained about like in microbiology techniques like bacterial and all. That guy is asking, his name is on Muhammad. He's asking what if there is a water contamination? Nimesh sir.
I am not getting exactly.
Is there some way for identification of viral contamination? He's asking like this.
Well, actually, pharmaceutical industries, we are not taking care of that viral contamination. We are more concentrated on that bacterial contamination only. Because
Thank you so much. Yes. Thanks for the answer. Hope on you satisfied in the answers. But I have one way, like if you want some information and things on viral contamination, you can do some sort of plaque tests by phase testing activity or plaque testing activity in your lab, and you can finish it at all. So the next question by Mr. Anup sir: what media is used in bioburden testing? Kind of media used in bioburden testing?
in bioburden testing, we are using soybean casein digestive agar.
Okay.
SCDA.
Okay, and next one by can I get PDF, PPT material and recording of the respective webinar? Yes, Vijay, I will share. This webinar is also live on YouTube. You can access the webinar video there on YouTube, and even if you share your complete profile, and definitely you will receive the recording of the session. So the next question by Mr. Hemant K. Gautam: how to do the room sterilization and how frequently?
By using the conventional method, loop sterilization is doing by using Bunsen burner or some electric heater is available. But instead of that, in current scenario, ready to use disposable loop are available. So it is advisable to that ready to use loop. So there was a less possibility of any contamination and everything.
Okay. Thank you so much. Thank you so much, sir, for this insight and answer. Hope you satisfied with the answer. Gopal Sabi is asking informative session. Thank you so much, Gopal, for joining us today. Sir, one more cross question by Hemant, sir. He's asking what is the latest technique? No, I mean in room sterilization. Is there some latest technology in room sterilization?
What?
Is asking like, is there some latest technique in room sterilization?
It's a hydrogen peroxide vapor phase technology. It's a dry fogging technology.
Okay, thank you, sir. And again, one more question. Can you explain? Ishwarya is asking, can you explain more about gel clot assay?
Technology?
Can you explain about gel clot assay?
Gel clot assay.
Yeah.
Yeah, okay. So in gel clot assay, there was one basic principle. There was one kinetic reaction was there in presence of that endotoxin. There was factor C is there. So factor C is converted into active factor C. So once the factor C is activated, proagulogen enzyme is there. So it is converted into clotting enzyme, and that's why the gel formation was occur.
Okay. And next is asking by Pratik, and he's asking like, what is the frequency criteria for area qualification?
Criteria.
Frequency criteria for area qualification. I think he's
Can you
asking
repeat,
something
please?
about on environmental monitoring, I think, sir.
Yeah. Can you repeat, please, question?
What is the frequency criteria for area qualification?
Frequency.
Sorry, frequency.
Frequency, na?
Yeah,
Yes.
frequency criteria for area qualification.
Frequency.
Okay, got it. There was no guidance on that, but what is the basic industry are follow? At 3 days, they are doing at rest monitoring, followed by 7 day at operation monitoring. And regarding that limit, whatever the pharmaceutical limit was given, that limit we are utilizing with that.
Okay, so sir, one more question is asking by Shiv Shankaran sir. As per WHO GMP fungal plates, as per the WHO, GMP fungal plates are incubated at 20 to 25 Celsius for 5-7 days. In this case, if I observe the plates on 5th days and release it, make any deviation to procedure, and how can it is justified? Any validation study required for minimum date for release?
Actually, 5 to 7 days. 5 is the minimum incubation time, and 7 is the maximum incubation time. So, for example, the fungal is there, and then it can grow within 3 days, because we already proving by the growth GPT plate, and we challenging that aspergillus niger in the SDA plates also. So, as such, there was no study requirement. And yeah, we can release the plate at minimum incubation time, that is the 5 day.
Okay. Thank you so much, sir. Thanks for answer. Host is Shankaran sir. You satisfied with the answer? The next question is asking by Sagar. Is asking how we determine water sampling point and sampling frequency.
Okay. So water sampling point actually determined by based on the usage of water. For what activity we need to utilize that water. So in that way we are designing. So during the design qualification of the area, we are giving that sampling point for different activity like granulation, coating solution preparation, as extra. So in that way we are selecting that sampling points. So strategy, what we can do, initially up to 1 year we do the extensive monitoring, and based on some data we rationalize and we reduce that frequency. So in that way we monitoring the system and we finalize the sample points.
Okay, thank you so much. Hope Sagar is satisfied with the answer. The next question is from Pratik sir. He is asking, is there any ratio validation required for the microorganism testing? Likewise, 2 years or 5 years?
Regarding method validation?
Yeah.
Actually, method validation we need to perform at initial level only, and later on, suppose there was some change in the methodology, some change in that manufacturing process. In that condition, we can repeat that validation. Otherwise, if no change, then no repetition required.
Thank you. Thanks, sir, for the answer. Thank you so much, Pratik, sir, for raising your question, and hope you're satisfied with the answer. Sir, one more question here from Sagar Kulkarni is asking, what is the best process for transitioning from one alternate bacterial endotoxin test method to another?
By transitioning, we need by using method validation.
Sagar
We
Kulkarni,
need
the best method is method validation for your question.
Okay.
Yeah, sir, is there one more question from Hira Chand Khade, sir? Sir, what is the difference between aseptic and sterile?
Aseptic is a practice, and sterile means free from microorganism.
Okay. Thank you. Thank you so much, Nimesh sir, for the answer. Hope Hira Chand sir, you satisfied with the answer. Dear guys, if you have any more questions and clarification regarding the session, you can simply text it here. We'll convey your question with the speaker.
Hi, good evening to all. Dr. Jitendra, can I ask certain questions on behalf of Abdos? This is
You can
Saurabh
ask.
Joshi, Preservative Product
Yeah,
India.
sir. Yeah, you
Okay.
can ask.
First of all, it was a great session by Mr. Nimesh. Thanks to him that we got the insight regarding all these techniques in the microbiology department, specifically QC. We have prepared 2-3 questions on the behalf of audience, on the behalf of our team as well. The aim of those questions is to get closer look how pharmaceutical or this pharma industry perceives the manufacturing, how they perceive the from manufacturing side, how we should tap into those things. So from manufacturer's point of view, like Abdos, so Abdos is basically into most of the plasticware products right now. So my first question is that how does this sterility assurance level certificate impact the entry of the sterilized product into a pharma industry? That is my first questions to Mr. Nimesh. What is the impact of that?
So for that, what you can do, you need to perform that first of all gamma radiation. So whatever the gamma radiation you are performing, you have that validated load, and after that, once the gamma radiation was completed, you need to perform that sterility test of basic components, and based on gamma dose and based on sterility results, we can assure that sterility assurance of that product.
Sure, because as a techno-commercial person, some manufacturers provide like sterility assurance level of 10 to the power minus 6. Some provide 10 to the power minus 3. So that was the, I think Annette has been there. Hello, can you hear me?
Yes.
Hello.
So that I can explain only. What for gamma radiation, whatever you are validating the dosing. So during validation, you are challenging the geobacillus thermophilus microorganism. At that time, you are challenging 10 to the power 4 or 10 to the power 6. And based on that, you are selecting the dosing condition. Whatever the challenge you are doing, that is your sterility assurance.
Okay. Okay, got it. Another question is like that. What are the parameters that one keeps in mind while adopting the particular pharmacopic guidelines in a pharmaceutical industry?
Parameter like first of all, you are coming as a sterile, then you need to perform the sterility. Sterility is a parameter.
Nimesh, can you hear us?
Huh? Hello?
Hello.
Yeah, Saurabh sir, he's answering your question, sir. We can hear him. Nimesh sir, proceed.
Considering the parameters, you need to give the parameter like sterility, bacterial endotoxin test result, Microbiology perspective view, it's a two parameter is most important. For non-sterile product, you need to claim what is the number of CFU present in your products, and gamma radiation certificates is for sterility assurance purpose.
Okay.
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 Nimish sir, sorry, Saurabh, sorry to interrupt you. There is one question from audience side. His name is Ranjan. Nimish sir is asking what is the difference between treated and untreated plates? Treated and untreated plates.
Not getting actually.
What is the difference between treated and untreated plates?
Plates.
Yeah, plates.
I need to confirm, and I will come back to you on that question, because I think so. Manufacturer is the best answer on the question.
Dr. Jiten, can I answer that one?
Yeah, you can proceed it now.
Can I answer to that question which Mr. Ranjan asked?
Yes, please answer, Saurabh.
Okay. Ranjan, the question is right now in this perspective is wrong, because there are no treated plates utilized in QC microbiology department. These are concerned with the cell culture products, and those treated and non-treatment depends upon the type of culture you are about to do regarding the culturing of adherent cells or non-adherent cells. So that is a different vertical you are talking about right now. It is not relevant to this QC microbiology. Okay, Mr. Nimish, last question from our side. Just a basic practical point of view, because you are definitely into pour plate and spread plate techniques, all these things. So we want to know some basic solutions to prevent the condensation inside the top of a lid after media is being poured. And at the same time, in some cases, solution to prevent the evaporation when it happens. The concerns which clients face.
Yeah, correct. So in that condition, what we can do, first of all, whatever the media, it's a melted and cool one. First of all, we need to cool the media up to 50 degrees centigrade. And meanwhile, we can start the preparation of the product.
Hello.
So in that condition, we are taking that product solution. We just open the half lid of the battery plate, inoculate the products. Meanwhile, temperature melting, media temperature should achieve 45 degrees centigrade. So around 45 degrees centigrade, you can pour the media in the battery plate, and you just close down it and solidify it. And from microbiology perspective, I recommended we can use that vented plate. So during due to ventilations, there was a less chance of that water droplets and everything. So actually, the precautions thing is temperature, media temperature should be not more than 45 degrees centigrade, and inoculum, whatever the product you are inoculate in the battery plate, so that time duration is very short, so we can solve both the problems at a time. We can manage the activity.
Okay. And Mr. Nimesh,
Thank you so much.
This question. Okay.
Hello.
Okay. Thank you, Dr. Jiten. Thank you, Mr. Nimesh.
Yeah, one more question here is I have received from audience side. So please listen. Nimesh sir, please be here only for next few minutes. The question is from Avinash Nawale. He is asking, is there any requirement for testing of BCC in finished product of API?
Requirement of?
Is there any requirement for testing of BCC in finished product of API?
ECC.
BCC.
BCC. I'm not getting what is BCC.
Avinash, can you please elaborate your question if possible? And he's asking one more question, sir. As per guidelines, what will be exposure time in different classes of area during plate exposure?
For that, you need to validate first of all, but basic industrial practice was 4 hours only. Could be not more than 4 hours. But whatever the 4 hour you are claiming, you need to validate it.
Thank you so much. Thank you so much for the answer. Hope Avinash sir, you satisfied with the answer. And if you need any more clarification, like any of your workflow issue or any of your consumables issue, contamination issue, and any of your microbiology techniques, you can simply write to mail ID mentioned in a chat box and Q&A box here, either to Saurabh or to me. We'll forward your question to Abdos guy, and you will definitely get the answer and clarification from the team of Abdos. And now, without further ado, we'd like to over this mic to Bijoy sir.
Sir, thank you very much for your informative session. That was really very informative. We'd like to just take you through the audience through a small PPT on some of the products that may be very relevant in the pharma industry. So we have centrifugal and microcentrifuge tubes, the freezing tubes, cryo vials, screw cap vials.
Sorry, Bijoy sir, I'm not able to see your screen. So before showing, can you please share your screen first?
Yeah.
Yeah, fine. Now you can proceed, sir.
Right. So these are some of the products which are relevant in the pharma industry. Centrifuge and microcentrifuge tubes, cryo vials, freezing tubes, screw cap vials, liquid handling using pipettes, pipette tips, serological pipettes, petri dishes, spreaders, tubes, test tubes, sample containers, bottles and carboys of various sizes, measuring cylinders as well as beakers, instruments like centrifuge range, microcentrifuge, plate centrifuge, microplate mixer, digital magnetic stirrer and hot plate, vacuum pump, of course, vortex mixer, digital dry bath, and digital incubator. So these are some of the products what we can offer from Abdos range for pharmaceutical QC. Thank you so much. Nimesh sir, thank you so much for a wonderful session.
Thank you so much.
Thank you, Microbios and the Abdos, to give me the opportunity to present here. Thanks a lot. Thank you.
Thank you so much, Nimesh sir. We'll be in touch with you for many more activity like this. And thank you all the attendees today who has given your valuable time for today's presentation. And really, I thought it was a wonderful session presented by Nimesh sir and Gaurav as a product insights of Abdos Life Sciences. So, Saurabh, are you there? Would like to say something in note for our audience today?
Yeah, definitely. It was fabulous session, and we had good insight from pharma industry as well. So we will every time we'll catch up. We'll try to innovate. We'll try to bring good things to pharma industry, to our end users, and definitely the audience who are aspiring to be in the pharma industry or to be on the manufacturer side regarding much bigger roles. I mean, everybody is trying to understand where one can exploit and one can use their talent in the upcoming time. It's a great session. Thank you, Microbios, Dr. Jitendra. Thank you, Mr. Dinesh, and thank you everybody for being part of this great, wonderful session. Thank you.
Yes. Before leaving the session, if Nimesh sir, are you there? There is one last question asking by one audience. That is Sunita. She is asking, hello sir, I want to ask in raw material sample. Do we get count without dilution factor?
We
Do we get some count without any dilution factor?
We can.
I think, sir,
We can give the count without dilution factor. Suppose you are analyze the product without dilution, then you can give the count directly without dilution factor only.
Okay. Thank you so much. Thank you, Sunita, for raising your question. Thank you, Nimesh sir, for the answer. Thank you so much, Marian, for giving your valuable time for today's session. Thank you, Himanshu, for giving these insights. Kailash Chandra sir, yeah, definitely you will get the link after the session. And I think, is it? Thank you all guys for joining us today. So,
Thank you, Dr. Mr. Nimesh, and thank you, Dr. Jiten.
thank you so much, sir. Thank you so much. So, can we go now, no?